you and Brian and Dane Wittrup and Jennifer Cochran and the other organizers for inviting me here today to tell you about some of our work. My lab is interested in the chemical biology of proteins, and that's what this collage means to show, some of the scenes from the kinds of projects that go on in my group. Today, though, I'm going to focus on one particular project, which relates to scenes from this bottom left, especially about the cytosolic delivery of proteins into cells. And the problem is basically summarized with this slide. Small molecule drugs represent about 80% of the pharmacopoeia. The targets for those drugs are roughly evenly divided between cell surface targets and intracellular targets. Of course, this includes GPCRs, and here we have kinase inhibitors and targets that exist in the nucleus of cells as well. The difference, as everyone in this audience knows, between these small molecules and proteins is that proteins act exclusively outside of cells. The antibodies that are the 7 of the top 10 drugs have epitopes that are on the cell surface. These proteins, of course, also include insulin and other hormones that bind to receptors on the cell surface and elicit signals that are manifested inside the cells, but the site of action is on the cell surface and not inside of cells. So what's the problem? Well, the problem is the lipid bilayer, of course, but also the fact that the cell surface is highly anionic, and I show you here a micrograph of the glycocalyx, which is composed of very anionic sugar moieties, as shown here, both sialic acid, which is often attached to gangliosides, as well as heparan sulfate and chondroitin sulfate. To overcome this, there have been many approaches. My group has been engaged in this for well over a decade. One of the approaches that we developed just over 10 years ago was to simply rely on Coulomb's law and say, well, if we want to get a protein inside the cell and the negative charges are a problem, perhaps we should use mutagenesis to replace negatively charged residues with positively charged residues. So what we did was to take GFP, which is what's shown here, and replace, using mutagenesis, 5 aspartates and glutamates with arginine residues. So by grafting on those arginines, we basically converted this anionic patch, shown here in red, into a cationic patch, shown here in blue. So this is the way that you could perhaps overcome that barrier using molecular biology. And this was successful in the sense that we can get GFP now to go into cells, but not in a desirable way. So here are some images of cells treated with unmodified GFP and then washed. The nuclei are stained blue with a Hoechst dye. And you can see essentially no green color here. The wild-type GFP doesn't go into cells. The supercharged GFP does go into cells, but this is a picture that we would consider in my group to be very ugly. It's very punctate. This GFP is getting driven into endosomes, into vesicles. Most of it will get destroyed in lysosomes downstream. This is not a desirable phenotype if you're interested in delivering molecules into the cytosol. There's very little druggable, for example, inside of endosomes. This is not a solution to the problem. In our experience, cationic peptides exhibit a similar phenotype. You can put a cationic peptide onto a protein and drive it into cells, but most of that material is also going to end up in endosomes for similar reasons. So we thought a few years ago about a different approach. And the approach really relies on biochemistry. This is the wall chart that was in my laboratory when I was a student. It probably not many of these exist, I think, anymore. But this is the primary metabolism that exists inside of all the cells in our bodies, as well as the bodies of other organisms as well, because so much of this is so fundamental. If you were to look at this chart closely, you would see that there are very, very few esters on this chart. The only esters that appear are the two that are shown here. Of course, lipids have ester moieties in them. The diacyl group here on on glycerol is made by esterification. But these esters, I would argue, are quite buried in the membrane and not especially accessible to the solvent, either outside the cell or in the cytosol. The other ester that's prevalent are the charged tRNAs that are used by the ribosome to translate proteins. So this is an amino acid that's attached to a large tRNA molecule, poised to bind to the ribosome and add on to the growing polypeptide chain. These aminoacyl tRNAs, though, are quite sterically, the ester groups are quite sterically hindered, and also they tend to be sequestered by binding to elongation factors, and so they're not readily accessible to solvents. The point is that these two esters can survive the environment in the cytosol, despite the fact that there are a lot of esterases in the cytosol and very high esterase activity. So that has allowed medicinal chemists for decades now to overcome that negatively charged glycocalyx that I talked about earlier. If there's a small molecule that has an anionic functionality, that anionic functionality can be masked in the form of a prodrug. The idea is to take this anionic functionality, like a carboxylic acid, most common, convert it into an ester here. That ester then is electronically neutral, can diffuse in and out of the cell. Once it gets inside the cell, esterases that are prevalent in the cytosol can convert that ester into carboxylic acid, recreating the active molecule inside the cell. So about 10% of small molecule drugs are delivered as prodrugs, and that number is increasing. In recent years, it has been actually over 20% of new drugs, new small molecule drugs being prodrugs. Here are some examples. These are some of these drugs are household names, quite well known, and I've just highlighted the ester moieties in them so you can see what I'm talking about. Most of these esters are simple ethyl esters, by the way, some isopropyl esters. The idea is that the hydrolysis of those esters leads to relatively non-toxic byproducts as alcohols. So what we sought to do was to ask this question: can we apply this very, very successful strategy of medicinal chemistry to proteins? Can we esterify proteins and get them to behave like small molecule drugs and cross the membrane? So if you wanted to esterify a protein or a carboxylic acid group in general, what you'd probably do is think about activating it with some electron withdrawing group to get this active acyl group here, and then attacking with an alcohol. And I put in here actually I put this in here, but I also include in this kind of paradigm simple Fischer esterification, perhaps the oldest way to make esters, involving concentrated acid to activate the leaving group. The problem with doing this, of course, and this I'll come back to this later when I'm going to talk about this electron withdrawing group being a carbodiimide. The problem with doing this is that this has to be done in water. And so water is going to be a vicious competitor for this alcohol and trapping that electron withdrawing group, and not allow you to get appreciable yield of an ester with this kind of strategy. So I call this an electrophilic mechanism because we're essentially making the carboxyl group into an electrophile that we attack with an alcohol. So this doesn't work. So what we did was to think about other ways that esters can be made, and we were led to Deazo compounds. So Deazo compounds, it was shown by actually Hermann Staudinger slightly over 100 years ago, can mediate the esterification of carboxyl groups by a mechanism shown here. And this mechanism was elaborated by Jack Roberts when he was a faculty member at MIT before he moved to Caltech. So the mechanism involves the Deazo compound shown here using the basicity of this carbon to abstract a proton from a carboxylic acid group to give rise to this diazonium species and a carboxylate, perhaps trapped in the same solvent cage. And in fact, that's what Roberts proposed way back in 1951. Then an SN2 reaction eliminates nitrogen, one of the world's best leaving groups, to give rise to the ester. So that's what we'd like to do, but there's going to be a problem with this as well. Water is a vicious competitor here because water can also react with a Deazo compound. So this Deazo compound can abstract a proton from water as well as from this acid, give rise to this diazonium species with now hydroxide as the ion pair, and then give rise to an alcohol. That's, of course, undesirable. We want the upper pathway. But we had, we thought, a window here because, after all, carboxylic acids are far more acidic than is water. We had about 10 pKa window here we thought we could hit by perhaps properly tuning the basicity of that carbanion there to remove this proton, but not that one. And so I'm going to cut through a lot of chemistry here and just show you the results by telling you, in the interest of time, that this is our favorite compound right now. So this diazo group is decorated on the left-hand side here by this toluene moiety. So the electron density, the negative charge on that carbon, resonates into this phenyl ring. We installed this methyl group here based on a Hammett analysis that told us we needed to have a little bit of electron donation to make this a little bit more basic, this carbon a little bit more basic to be optimal. On the right-hand side here, we have an amidic moiety. And I'm showing you the dimethylamide because I'm going to emphasize today cell permeability. But of course, there's a lot of variability here, and these can be replaced with targeting agents, and that's something we're working on as well. So this compound gives rise to efficient esterification of carboxyl groups of all sorts in water. And I tell you, too, that it has good Lipinski properties, if you will. It has no hydrogen bond donors, only one hydrogen bond acceptor, and it increases the log P of a carboxylate group by 4 whole units, which is 10,000-fold. And the chemistry is described in these papers that I'm not going through in detail. So where we are right now is the thought that maybe we can take a diazo compound like this, use it to esterify carboxyl groups on a protein using the Roberts mechanism shown here, take the resulting ester, hope that it goes through into the cytosol where it will get hydrolyzed by esterases just like a small molecule drug. Just to show you some data, this is a typical QTOF mass spectrum of what happens when we treat GFP with that diazo compound I just showed you. Here's where the native GFP would be, the wild type enzyme. So by using that diazo compound, we're adding approximately 10 plus or minus 1 or 2 ester functionalities to the GFP. Which carboxylic acids get modified? Well, we've done some MS analysis on this, and I can tell you that it follows what you'd expect from a physical organic chemistry point of view. The carboxyl groups that have the highest pKa, which need to follow the Roberts mechanism or can follow the Roberts mechanism most easily, are the ones, the filled circles here, that tend to get esterified. And also we hit more glutamates than aspartates consistent with steric factors. We don't hit buried groups, however, as you'd expect, because we're not denaturing the protein. So here's where we are right now in this talk. This is GFP, the wild type enzyme. It has 32 carboxyl groups, a net charge of -9. That's lysine plus arginine minus aspartate minus glutamate. When we esterify, it looks something like this. This is a model of what it looks like. So we've wiped out some of the red patches and added some white patches to give a net charge of +1. Now we have 21 carboxyl groups instead of 32. So let me show you some key fact here, which is that this is all bioreversible. This is just like a small molecule prodrug in the sense that the esterases in either cell extracts or in whole cells, we fish these molecules out of cells. All the esters are removed by the esterases that exist inside the cell. So that means we're not going to compromise structure function or generate immunogenicity. We've also tested this alcohol, and it's not toxic. Some data. So this is wild type GFP, unmodified GFP, and these are just different channels here. This is, again, the nuclei stained with a Hoechst dye. This is a wheat germ agglutinin stain to see where the cells are, the outside the cells. After washing, there's no wild type GFP that enters cells. Here's the merged image. The image is very different when we esterify the GFP and expose it to cells. Now we see lots of green inside the cells, and it's not punctate. I'll show you a blow up in a moment. It's very diffuse staining. This is perhaps the most interesting panel here because this is what happens at 4 degrees centigrade. So at 4 degrees centigrade, it's well known that there's no endocytosis into cells. You can't form endosomes. You don't get vesicular localization. Yet we get the same image here as we do here, which suggests that endosomes are irrelevant to our translocation process. Here's a blow-up of that 4-degree panel. You can see the staining is nicely diffuse here. The brighter images are just closer to the lens of the camera, and the darker ones are farther away, but we don't see any, we see that all cells take this up and take it up in a very diffuse manner. Here's that supercharged GFP again, now at 4 degrees. Remember that ugly picture I showed you earlier? Now you don't get any uptake because, again, that has to go through endocytosis, so the cells don't accumulate any green color at all at 4 degrees centigrade. So this is a marked contrast between these two methods, the chemistry on the left and the molecular biology on the right. We've put a nuclear localization signal onto the GFP to try to drive it into the nucleus. That makes the protein markedly more cationic, so it sticks to the outside of cells even after washing without esterification. But when we esterify the protein, now we can drive it into the nucleus, and there's significantly more green color in the nuclei here than without the nuclear localization signal. So we think we can drive proteins into the nucleus using this technology as well. In unpublished work, we've done this with a Fab fragment. So this Fab fragment was labeled with a psi dye, so it's purple. And you can see the unmodified dye doesn't go into cells at all. The unmodified, Fab fragment doesn't go in, but the esterified Fab fragment goes into cells. It goes into the nuclei, actually, specifically, and we were puzzled by this at first until we looked at the sequence and saw that it had its own internal natural nuclear localization signal, something we didn't expect until we got these images. We've recently shown that we can get an enzyme to go into cells and that it's active once it's there using this technology. So wild-type human ribonuclease will not go into cells efficiently, but when we esterify it, it does go into cells, and not only that, it kills HeLa cells because of its ribonucleolytic activity. This is essentially a titration curve because human cells have an endogenous ribonuclease inhibitor protein that binds to ribonuclease 1 with femtomolar affinity and is present at low micromolar concentration. So what you're essentially doing here is just titrating out that inhibitor protein, and once you do that, then the ribonucleases are free to cleave cellular RNA and kill the cell. As a control for this, what we did was to eviscerate the active site, change 3 key catalytic residues to alanine, so this is an inactive catalyst, and now whether it's modified or unmodified, it doesn't kill the cell. So these and other data suggest to us that in fact the enzyme is active inside of cells. This article is just out online now. Okay, so last few slides, you're probably wondering about the mechanism of how all this works. We have some data on that. I'll tell you about that now. The question we've been asking is ester hydrolysis necessary for cytosolic entry? And as a test of this, what we did was to make the same kind of molecule I've been showing you, labeled GFP, but labeled with an amide linkage instead of an ester linkage. So we did this by taking GFP, and it has, of course, these 32 carboxyl groups, and then reacting it with the same molecule I've been showing you, but not as a Deazo compound, instead as an amine, where we use a carbodiimide, a water-soluble carbodiimide, to do the coupling reaction. So this is well-known chemistry. We didn't invent this. It's very much standard procedure, but we can make this way GFP that's decorated with amides instead of esters. And so what we did was to do the same kind of experiments I've been showing you and ask whether this goes across the cytosolic membrane the same way the esterified GFP does. And the answer is it does not. It goes into endosomes. What we've done here is, of course, make the protein more cationic, and so we get these kind of ugly punctate pictures, not unlike what I showed you earlier with the supercharged GFP. So this is being driven into endosomes at 37 degrees. And at 4 degrees centigrade, it doesn't go into cells at all. It's just sticking to the cell surface, just like that GFP that's labeled with a nuclear localization signal. It's much more cationic now. It's hard to wash off, but it's just sticking to the cell surface. This is a far cry from that uniformly green-colored image that I showed you earlier. So what does this mean? Well, this is our model for how this works. We view the protein as kind of like a giant iceberg. This is outside the cell. It's starting out here. It's coming to the cell surface, the interface between the inside and the outside, which is a lipid bilayer, like this air-water interface. A little bit is poking through to the inside, we think. We've coated this cell. We've not only gotten rid of the negative charges, or a lot of them, but we've also coated it with this rather hydrophobic moiety, this phenyl group, this good Lipinski compound. So we think that this can reside in the membrane to some extent. Here's a to-scale cartoon of what I'm talking about. This is GFP shown with a lipid bilayer here and an ester moiety poking through to the inside of the cell into the cytosol. What we think happens is that ester moiety encounters a lipase or esterase that generates a negative charge. That negative charge then is going to be repelled by the lipid bilayer on the inside of the cell. That's going to generate a force that's going to pull it inside the cell. So this is different than the mechanism by which prodrugs enter cells. That's a passive mechanism. Here we're actually requiring the hydrolysis of the ester moiety to drive the process. So we're not aware of biology or even chemical biologists using this kind of mechanism previously, but all our data suggests that this is the way this is working. So with that, I'd like to thank you again, thank the organizers again, and thank the people who are doing this work. This is the group of people in my lab right now who are working on this project. We have wonderful collaborators, including my friend and colleague Dane Wittrup at MIT, and also Jim Wells at UCSF, who supplied that fab fragment for us. These are the people who did the work that led us to the current stage, and this is a recent photograph of my group having a good time at a Boston Bruins game not too far from here a couple months ago. And with that, I'd like to thank you, and I'd be happy to answer any questions.